So how does the actual release work? There's a big protein complex called SNARE on the outer membrane. And in the vesicle, there is another protein called synaptomogen. Not really sure how to pronounce that. And it's calcium sensitive. So again, the action potential reaches the end and calcium flows into the cell. So this reacts with calcium and docks with SNARE, kind of locking it in place. And then a structural change happens to SNARE, causing the membranes to fuse together. And when they fuse together, this membrane becomes part of this membrane, and it releases everything inside the vesicle. So, and it also doesn't happen all at once. So there's all these vesicles waiting for the action potential to arrive. And they actually release in quantals. So you can see here this is looking at the rate that they're being released, and it kind of peaks and dips and peaks again and in these steps. And this is because you can only have so much area that the vesicles can release on. So first, again, you have the influx of calcium from the action potential, and it causes the vesicle to bind, and you can see the SNARE right here. And it binds and it releases its neurotransmitter, which goes across to the postsynaptic cell. But then you also have these vesicles that are just kind of hanging out, waiting their turn to dock. And so what's happening here is a whole bunch of them are docking and releasing, and then they have to redock. The new ones have to come in and dock, and then they release. And then some new ones come in again and again until the release is done. And there's about 200 vesicles are released per action potential at a neuromuscular junction, where in the central nervous system it might be 1-to-1 ratio. And this also is another mechanism to regulate the response. Because if you have a lot of neurotransmitter going through, you're more likely to get a response than one neurotransmitter or one particular neurotransmitter. So there's 3 types of vesicles. So there's the primary ones that are docked and ready to go. There's the ones that are waiting for their turn to replace the ones that were just released. And sometimes you have things called dense vesicles. And these aren't filled with neurotransmitter. They're filled with proteins that help modulate the synapse in various ways, either by docking to various other proteins in the membrane, or going across and maybe blocking something, or doing or opening another channel so that it can accept a neurotransmitter, or various other things that would need proteins at the synapse.